This lecture is being brought to you in part by the generous gifts of these sponsors.
Good evening, everyone. Thank you for coming. And thank you, Bonnie, for that wonderful introduction. Since this is a broad audience, which I don't usually get to talk to as much in our academic environment that I live in, I thought I'd make this a little more personable with this lecture by doing two things. One, I want to give you a little bit of a background into viruses and vaccine development over the years and why this is important and how our contributions fit into that larger picture. But I also, just for fun, to ground myself in my day-to-day life of doing experiments and talking to students, one thing I like to do is I collect quotes. It kind of keeps me grounded, and I thought just for fun I'd sprinkle a few quotes in as I went from my collection, give you a sense of what I'm like. And I just want to start with kind of a broad quote, and that is, "We make a living by what we get, we make a life by what we give." Now, some of you may recognize that quote, and I'm not going to quiz anybody, but if you know the quotes, authors, you can pat yourself on the back as we go. But that is by Winston Churchill, and the reason I bring this up is because it's important to remember not just the broad, it's a quote to live by, but it's also just a grounding quote in the sense of, as a scientist, what do we do? And what we do is we try to get results. And we try to get results then that, I guess, as an academic exercise, contributes to human knowledge. But once in a while, we get to do something a little bit more, and it's something that benefits society more directly. And that's one of the things I want to talk to you about today that we're doing in our laboratory. It's also, I think, important just to keep this in mind from the perspective of, it's important to give back in a more direct way. And having the opportunity to talk to you, and for all you coming, and for Dr. Ken Ford for giving me this opportunity, I'd like to thank everyone. The second quote I just want to start with is now delving into the science of virology. And this is kind of a dark but poetic quote, so you know it's not a new one. The most dangerous epidemic is the smallpox, which sweeps at times like a storm of death over the land. So this is from the mid-1800s. It's by Sir Richard Burton, who was an explorer and traveler and a Renaissance man, had many specialties. But while traveling through Asia, he saw firsthand variola major, which is a severe form of smallpox. People who contracted it typically only had about a 50% chance of surviving the infection. And it just adds perspective because we sometimes forget what life was like before we had some of the scientific discoveries that have been at our disposal. And when our world is much different now as a result of what has happened with the development of approaches to eradicating some of these diseases. So variolation was a process in which you could take a smallpox lesion from an individual, collect it, and then give it to somebody who hasn't yet been infected. And the idea was, in Asia, that when the major form, which was so lethal, was sweeping across the land, if you could give them the minor form, which was far less lethal, intentionally, you could protect them before they got hit by the nasty version of it. And so that was called variolation. There was, in Europe and America, the smallpox that was going around was mostly the minor form. And so it wasn't as lethal, but if you did get it, you would get pox lesions typically around your face 80% of the time. And these lesions don't clear up well. They left you permanently scarred. And so it was disfiguring, and it was a very terrible thing from that sense. And there's a figure from our history, Lady Mary Montagu, who was traveling abroad, and she saw some cultures locally doing this variolation process, and she brought it back to the Western culture and said, we can just cut under a person's arm or on their foot, and you could rub in this material and get an infection started that was a little bit more hidden away rather than letting it naturally get onto your face. And that would keep it under control and then get a nice protective response in your body that would prevent you from getting infected ever again. So that's all variolation. Now, some of you may know the story of this gentleman, Edward Jenner. He made an important observation that cow maidens who were milking cows were getting pox-like lesions on their extremities, on their hands and their arms. And these ones healed over very well. They didn't tend to leave scars. And the woman didn't seem to be suffering much other than just the appearance of these lesions. And he made the connection that this was a related virus coming from the cows, and that you could harvest this material instead of material from an infected human and use that to expose a person and get them immunized. And so since it was coming from a cow, the Latin word for cow, as many of you know, is vacca. And so that's where vaccination comes from. Oh, I guess a lot of you didn't know that. Now, of course, vaccination has a mixed reception often, and the reason is that you are intentionally giving something foreign to a person, and that can have adverse consequences, even though the intention is to benefit that individual, and it usually is quite beneficial, but sometimes things can go awry. Some people may be predisposed to be more susceptible to a pathogen that wouldn't cause any disease in a typical person. And so as vaccination was being done, there was raises of alarm. I've taken one notice from the I think it was 1880s. And this one basically is just simply saying that people are being killed by this, and it's wrong for the government to be enforcing us to have to get vaccinations to smallpox. And so it'd be over 100 years at the advent of this vaccine approach by Edward Jenner before we saw a major revolution. While that was going on, we also had in the 50s, that some of you may remember, polio. Now, polio started before that, and it's always been around, but in the 1950s in the United States, it really erupted and became a major outbreak. And what you're seeing here is an iron lung ward from that time. And many of you probably know this, but I'll just go through it, just make sure we're all on the same page. What you're seeing here are these horizontal contraptions where a person would lay in the chamber and only their head would stick out the front end. And the point is that when this virus, which I should take a step back and say this is a very simple virus, it's referred to as a fecal oral pathogen for reasons I think that are pretty self-explanatory. Just washing your hands is an amazing way to defeat spread of this virus. But it can, unlike many other fecal oral pathogens, on occasion cause a much more severe form of disease. It will get into your nervous system and ascend into your spinal cord and damage those nerves. And when that happens, you can get paralysis, and that is poliomyelitis. And if it ascends far enough, the muscles of your diaphragm will stop to work, and now you can't breathe for yourself. And so the iron lung ward was to provide a way for these people who cannot breathe for themselves to respirate and stay alive. And so it would provide pressure inside that chamber that would cause your diaphragm to raise and contract for you. And if you have good eyes, you may notice that a lot of the individuals in these iron lungs have next to their exposed heads balloons. And that's because poliomyelitis was striking the young more than it was just about anyone else in the demographic. And so there's a lot of kids in this iron lung ward. Some of them would later recover or partially recover, but some would be paralyzed for the rest of their lives. FDR started the March of Dimes. We all know his face is on the dime now, but back then it wasn't, and this is what he was doing. His proposal going back to the late 30s was if everybody could contribute a dime to the cause, we could develop a way to defeat polio in the United States and perhaps the world. So what's interesting about this period of time is that it caused a revolution to occur in the sciences. With the influx of money that the March of Dimes brought, basically the birth of modern virology was started. And a lot of things came out, a lot of important discoveries came out of this period, but one that I just want to emphasize in particular was the ability to take mammalian cells, human or animal, and culture them in a dish in a laboratory as a way to study the virus. Before, you would have to use an animal, and it was very complicated, cumbersome, and expensive. But now you could have these individual cells derived typically from a cancer and let them grow in a dish autonomously. And so this is just what it would look like. This is a picture taken in my lab of just a very simple microscope looking down at some cells in a culture dish, and you can see there's probably about 7 or 8 cells in that field of view. Now let me take a moment here to diverge and just tell you a little bit about the cell, because to understand virology, you have to understand the field of cells or cell biology, and I just want to give you a brief primer on this. The center of the cell is the nucleus, and so this is an enclosed off area, and you can think of this as the library of your existence. Everything that is you is in your genes, in your chromosomes, and that's all in the nucleus. And one way you can think about how the nucleus works is that you have a bunch of scribes, and they're in there, and they're looking at this library of information, and whatever is important for the moment, they copy it down, and they send it out of the nucleus into the rest of the cell. And these little messages are called messenger RNAs. It's DNA being transcribed into RNA, and they're constantly at work. Now, where do those messages go? Well, they go into the cytoplasm, and there's one of the most interesting molecular machines known to science. It's called the ribosome, and this is a machine that will read that message and turn it into a three-dimensional structure that can do chemistry, a protein or an enzyme. And I like to think of it as a 3D printer. But a 3D printer does much better than anything we currently have. Not only can it print whatever message has come out of the nucleus into a three-dimensional structure, like a gear in this particular case, but it can also make more copies of itself as needed. Now, when these cells are doing what they do and staying alive, and they have this process continuing on, everything's fine. But if they detect a virus, and they can do this because they have a number of protein sensors that were made on the 3D printer, both on the outside surface of the cell as well as inside, if they can detect that there's something foreign that's nothing like themselves, some pattern unique to that incoming infectious particle, an alarm can go off. A message can be sent to the nucleus, to that monk who's doing the transcribing, and they can say, drop everything you're doing, start transcribing other books that have genes and genetic information specifically for dealing with infection. And so now the 3D printer might be printing out hazard symbols saying there's an infection here, and it will send that symbol out all to the neighboring cells. So before the virus even has a chance to get out of that cell, all the neighboring cells are basically on the alert. There's an infection coming your way, get ready to deal with this. And that's a surprisingly successful strategy. We live in a sea of viruses. Most of them will never cause any damage to you because of this simple little routine that's always going on. But there are a few viruses that have become pathogenic that can defeat this. In the case of polio actually completely shuts down the nucleus of our cells. It closes all the doors and windows, and instead uses its own monk, its own transcriptase, to make its own messenger RNAs right in the cytoplasm of the cell. And so by shutting down the nucleus, your cells can no longer warn other cells. They can't even try to defend themselves to making more viruses. And the virus will now commandeer that 3D printer and tell it to only listen to it and its genes and make more proteins from that. And so the virus will start to replicate, and it will do it very efficiently. The long term, it's not going to win. Your body will ultimately be able to catch up and defeat this infection, but the damage can be done before that happens. So with that context, I just want to point out that from this March to Dimes research, Enders and his colleagues received the Nobel Prize for being able to not only culture cells, but then to use those cells to produce poliovirus. And they were able to produce the poliovirus because the poliovirus defeated the antiviral systems in the cells and allowed lots of viral particles to come out that could be then collected and used for study. And this is just what an experiment looks like in a lab from the time, and it's not that different than it is today. What you're seeing here is an infection. Oops, that's not the pointer. You're seeing an infection with different times, and at the 0 time point, what's happening in each case is the scientists have basically collected about 1 million cells in a dish, and they basically made a protein soup out of it. And they took all that protein and they separated it out by size by a process called chromatography. And in a healthy cell, there are so many different size proteins being made that it's just a big smear of different proteins, and they show up as these kind of horizontal little bands. But as the infection progresses, you can see that all the cellular material is starting to fade, and you're left now with a very specific limited set of proteins. And these are all poliovirus proteins. The virus has taken over the ability of the cell to produce proteins. It's turned it into a zombie cell, and it's a viral factory by 5 hours. Okay. So the lemonade from this is that we got a vaccine. Jonas Salk, using Ender's cell culture system to produce poliovirus, collected the virus. He then added formaldehyde to it to kill it, and then he introduced that material into our bodies. And our immune system responded, recognized it, produced an adaptive response that then protected us in case we ever got exposed again. And so that's the cover of Time from 1954. And some of you probably know that Albert Sabin also came up with a vaccine by a slightly different method that wasn't fixed, it wasn't dead, the virus was still alive, but it was now declawed. It was a very passive virus that would replicate. And the nice thing about that is you can now just inoculate kids with 2 drops of this infectious material, an infectious material that will spread in your body temporarily, but won't cause disease. And that was a very easy way, an inexpensive way to transport this around the world and vaccinate kids everywhere. And the World Health Organization is using this to try to completely eradicate this virus from humanity. And speaking of eradication, eventually the smallpox vaccine did catch up. And if many of you know, this was, I would argue, the greatest success ever of the life sciences, was the ability to come up with a vaccine to defeat smallpox and eradicate it completely from society. So there's the context of what is driving me, or at least one aspect of what we do in the laboratory. And you might be thinking, why would you want to break a détente with a virus? A détente should be a good thing. And let me show you why it's a complicated problem. So when you get exposed to a herpes virus, these neuroinvasive pathogens at first don't seem like they're infecting your nervous system at all. Instead, you get exposed at your body surface, like around your lips in the case of herpes simplex virus type 1. The virus propagates. You might get a lesion. If you're a child, it could present in the most dramatic way to present something like this, gingival stomatitis. It doesn't look pleasant, but it's really not a big problem in and of itself, and it will go away and fully clear without scarring. And then later in your life, suddenly you'll get a recurrence. And this time it may present as herpes labialis, or the common cold sore. Now, what's really going on is when you first became infected, the plot here, the curved blue line, indicates your viral load, meaning how much virus replication would be occurring in you at a given moment. And when underneath the curve you get this color, that's indicate that you have symptoms that people can see. You're presenting evidence that you're infected. And so the child starts to get the gingival stomatitis, but he was actually infectious prior to that, and perhaps even after that. And then later in his life would be the cold sore. But the fact of the matter is, most of the time, you don't present any symptoms at all during these recurrences. So given the size of this audience, I would say there is a very high probability that there is some herpes simplex virus in this room right now out on body surfaces, and none of us know it. In fact, this virus is so successful that in this age group, and I include myself with you, about 80% of us are infected by this virus. And as far as viruses go, that's about as successful as nature ever gets. Now, why is it so successful? It ties into this neuroinvasive property that I've been alluding to. So let me go through that infection one more time with this illustration. You get exposed to the virus, it enters in cells at the body surface and propagates. Then it sends progeny viral particles down nerves that innervate that area into a part of the peripheral nervous system. And this particular example would be part of your trigeminal ganglia. So if one of you don't like what I'm saying and you come up and you punch me in the face, the reason I'm feeling that is because a nerve conductance right down this axonal shaft was fired, which then propagated to a second order neuron in my central nervous system and to higher cognition. I said, ouch. The virus is using that same highway, but it's not using it as an electrical impulse. It's using it as a physical entity to transport, like a subway car, or like I like to call it, a marathon runner. And you'll see why I like that analogy. Once it gets there, the infection goes to sleep. This is a classic aspect of all herpes viruses, is their ability to establish what's referred to as a latent infection. And this is why you carry it for the rest of your life, and why there's no cure to these infections. It's in the nuclei of those neurons within that part of your peripheral nervous system. And although the immune system adapted and destroyed the infection and sterilized it out here, it never realizes that you have an infection in your nervous system. Later in your life, if you're young, you might be studying for your final exams. If you're older, it could be sunburn or a number of other things. The virus can become reactivated, usually associated with some compromisation of your immune system. And this is one thing that just really caught my attention when I was entering this field that fascinated me, is that these new progeny particles, which might be made a decade later from the original infection, are somehow designed to go the opposite direction than the original virus came in on, and that way get back to the body surface and amplify in those tissues, so now you can spread it to all your friends and family. Sometimes you can get complications from this relatively benign process. The virus can spread to the eye. Herpes simplex keratitis is one of the leading causes of infectious blindness in the industrialized world. And on rare instances, it can spread into the central nervous system, and that's very problematic. This can cause this is herpes simplex encephalitis. It's typically fatal. If a person is caught quickly and rushed to the hospital, they can be put onto antivirals that can save their lives. The prognosis, however, still will not be good because the virus will now be latent rather than just in the peripheral nervous system. It will now be latent in this individual's central nervous system. And when they get their next recurrence, instead of getting a cold sore, they'll get a second round of encephalitis, which is equally fatal to the primary infection of encephalitis. So it's a terrible outcome. Fortunately, the numbers are low. And we're starting to understand that the people who succumb to this much more severe form of infection by this pathogen actually have deficiencies in their immune response. And if you would like to hear more about that, I can talk afterwards. So most forms of disease, whether they're mild or severe, are dependent on the ability of this virus to move these long distances in your nerves. And so that's one of the processes that we've been studying in my laboratory. And I also want to point out at this stage that the herpes viruses are very unlike the other viruses I mentioned at the beginning of my talk with polio and smallpox, in that these are ancient pathogens. Ancient meaning that they're probably much older than we are evolutionarily, and they've been with us as we've been evolving from a primate ancestor all along, in fact, going even further back than that. And one interesting thing is that all of these different primates get infected by neurotropic herpes viruses. And if you look at the genetic coding of those viruses and say, how do they vary and which ones are more similar to other ones, you can make a relationship tree, a phylogenetic tree, much like this one is made up for these primates themselves, the host species, and it matches this pattern exactly. The implication is that the viruses have been co-evolving, and as these different animals have evolved and broken out, the viruses have been evolving with them this whole time. And so we're left with 3 as a result. Herpes simplex virus type 1 and herpes simplex virus type 2, and the causative agent of chickenpox and shingles, varicella zoster virus. HSV type 2, you may have seen in the news recently. There was a public interest story that made it to the common press that HSV2 in humans is genetically a chimpanzee virus. And the reason this was, and science has known this for a long time, we've had the sequences, but the reason it made press is because there was some indication in this recent research that we didn't get it from a chimp, but that back in history there were a lot of other hominids that don't exist anymore. And it was those hominids that seemed to be the intermediates that got it from the chimps and then passed it to Homo sapiens, to us. And so that's one exception where the tree doesn't perfectly match. It looks like that was a much more recent jump. So here are those viruses that I talked about that are infecting us in humans on a routine basis. There's one other virus, at least, that can infect us in this group. It's called monkey B or herpes B. It's actually an old world monkey virus, one of those other monkeys on that last chart. And the reason this is important to note is that when it gets into a person, it doesn't cause these fairly mild cold sores, but instead goes straight into a person's brain. It's 100% lethal. Fortunately, it's not something that we get exposed to very often. If you have a pet rhesus macaque, you want to make sure before you buy it that it doesn't have monkey B. Hopefully they're checking that for you. Also, the rhesus macaque may be worried because it turns out our herpes simplex virus type 1, if it goes to the rhesus macaque, will go straight to its brain and kill it. And so these viruses have evolved this détente with their host species so that they can survive in us for our lifespan without hurting us. And by default, they can hurt us, and our immune system can't stop it, just as this illustrates. So, from an evolutionary standpoint, the way science thinks about these kind of concepts is under the idea of selective advantage. If there was a selective advantage for this virus to kill us, it certainly could. But by killing us, it burns down its own home. And there's no advantage to doing that. And so the détente is basically issued by the virus to us, saying, we're all going to be happy together, just do what I tell you. You can tell I'm a virologist. Okay. So, with that kind of concept, let me tell you, and because of that, ancient origin and the fact that it has such a hold on us evolutionarily, the thought of being able to get rid of it has been challenging for the past 50 years. So let me show you what we're doing. So first, let me introduce you to the virus particle itself. This is an electron micrograph. So this is a very thin section of those cells that I showed you in that plastic dish. It was cut with a special type of knife, made very thin, and then put into an electron microscope, and you can see, and stained, so you can see some contrast. And so this is a viral particle on top of a cell. as if it was getting ready to either come in or just came out of that cell. And the structure, as you can see, is kind of concentric. What you can't tell from that simple image is that the inner core is actually a polyhedron. It's a perfect icosahedron, a 20-sided object. That's a hollow protein ball. Inside of there is DNA for the virus. Surrounding that is a layer of additional protein that is referred to as the tegument for wrapping. And then surrounding this layer of protein is a membrane, much like the membrane that a cell has surrounding it. And so sticking out of the membrane are a number of additional proteins that the virus uses to interact with cells and enter them. So those are the parts that I just told you. And let me show you something that I'm very excited about that is just being published. It's a collaborative work that I did with another laboratory. This is another electron micrograph of these viral particles. And the first thing you look at that is, that doesn't look very good. There's not very much contrast here. But this is using an advanced technique that you may have recently heard of, which I'll tell you in a second. And basically, what you see is we had purified a lot of these extracellular viral particles, and you can make out that they have membranes. But these ones are flash frozen at extremely low temperatures. And the reason you do that is it causes the particle to be preserved exactly as it would be in nature. It doesn't get deformed in the processing events. And you can't really make out from the initial image, but because they are so well preserved, you can take a picture of this and a picture of that one, and in fact, a picture of 100,000 more. And you can take all those pictures and put them together and average out the picture and get a sense of what that virus looks like. And by having that average map, you actually get a much higher resolution, a detailed look at it than you could ever achieve otherwise. And this breakthrough just got awarded the Nobel Prize in chemistry. Not specifically for viruses, but just in a general sense. It's called cryo-electron microscopy. And science has now been using this approach to study proteins in great detail and large protein structures, including viruses. And so in the work that I did with a colleague, Hong Zhu, in California at UCLA, we came up with this beautiful, detailed imaging of the central core polyhedral capsid of the virus. And I just think it's really gorgeous. And what's also cool about this is you get so much detail that you can map this out in 3 dimensions. Now, one thing we're learning about this virus, and look at the detail you get out of this. It's just amazing. It's amazing. It's like the Death Star. So right there is a little piece now taken out just through the computer processing and just showing the detail level of individual proteins that are now been artificially color-coded. Each color is a separate protein that was made on one of those 3D printers and put together into this larger order structure to make this very large complex. But remember, this large complex was just a little tiny part of the overall viral capsid, that polyhedron. But it's a very interesting area, and particularly these parallel strands up here turn out to be where all the action's happening that make this capsid dynamic inside cells. You can think of this capsid, this huge polyhedron, as a big oil tanker coming into port. It can't really do much on its own. You need the tugboats to come in and push it around. And this is where everything's going to dock and all the excitement's going to happen. Okay, so the virus particle is a nanomachine. As it comes into a cell, it makes contact with those glycoproteins I mentioned. It then binds to a very specific receptor, which triggers an event that allows its membrane and the cellular membrane to fuse together. In so doing, the capsid in the inside, that polyhedron, gets deposited into the cell, along with those individual tegument proteins, which I had highlighted in green earlier. The capsid now needs to get to the nucleus because the genetic content is going to be transcribed by that monk inside the cell. Not all viruses do that. Polio does it all on its own in the cytoplasm, but this particular virus, this ancient virus, is using the nucleus for this purpose. So the monk gets at it, he sees this information, says, okay, I'll copy that. And he sends off the new messages out to the ribosomes. The ribosomes start making those individual proteins. They get brought back into the nucleus, and they assemble into more viral particles, more of these large polyhedrons. And so that's how the virus is going to replicate. And so how do we work with this, and how do we study it? There are 2 things in our laboratory that we developed that have given us the opportunity to really understand the mechanisms by which this virus infects us and how we can stop it. So once you have the herpes virus, you can isolate its DNA from its core. So that's just what this represents. We call that a plasmid in the lab. It's got 160,000 nucleotides or base pairs in it. That's the code. And we've cloned this into bacteria called E. coli. Some of you probably heard this. This is the bacteria that people in research labs use for all of their abilities to manipulate DNA. And by getting this cloned as a large plasmid E. coli, we basically now had a keyboard in front of us where we could start typing in code into this plasmid and then reintroducing it into cells after it's been modified. So for instance, I might put in code to make a fluorescent protein encoded within the genome. A fluorescent protein will glow in the dark when excited with the correct wavelength of light. When you put it into cells, the virus will start to be made. All those proteins are made. They'll self-assemble into new viral particles that are glowing red, and they'll continue to spread from cell to cell and propagate. Unlike poliovirus, this virus can defeat all of the antiviral tactics our cells have, and that's why it's always going to win and why it's so easy to make lots of virus in culture. So another quote, it must be explained, for example, why a virus causing no more than a cold sore in one person can produce fatal encephalitis in another. 1968, a year after I was born, and I think a quote that still carries much meaning today. We still don't have a complete answer to this question. But we can now take these red fluorescent viruses, so we made them glow in the dark. These are individual viral particles looked at in a microscope. And I should point out that the particle itself in this illustration is only about 200 nanometers across. That is so small that you can't even see it through a standard microscope. But you can see this little blip of light that it's giving off as a result of putting the fluorescence into it. Kind of like you see a star in the sky. You can't make out the star's surface, but you can see the light that it's projecting to your eye. And so we can see this light to indicate where it is. And what's nice about that is now instead of just looking at electron microscopy where everything's been fixed and it's dead, you can look at it in a living system. Ideally, you would like to see how the virus gets into the nervous system. And so you'd like to see that virus transmitting right down those nerve fibers. None of my graduate students have been willing to let me do this yet with them. I keep asking. But you can culture neurons in the dish. And so these are nerve fibers that have grown out from a central ball of nervous tissue. And you can image this just after adding the virus. Now here's a traditional fixed sample on an electron micrograph, very high resolution. These are the sides of a single nerve fiber, an axon. Inside you can see these like ropes. They look like ropes, and these are called cytoskeletal filaments. The inside of a cell to anything, such as a virus or a ribosome, is basically like being in space. There's nothing to get you anywhere. You're floating around. Gravity has no meaning at this level. And so for it to move somewhere, it's got to be able to have some way to grab onto something and pull itself. And so these ropes, called microtubules, for the aficionados in the audience, are a track that these viruses can grab onto and move. And I should say, it's not exactly like the vacuum of space to an astronaut. It's more like an astronaut that's in maple syrup. Because the cytoplasm is so thick that you're really stuck in quicksand. And so the virus basically has to pull itself forward, hand over hand, along these ropes to go anywhere. And it uses a cellular machine to do that called dyning. And so what I've told you so far is that the virus enters into the cell, it binds, I actually haven't told you this part yet, it binds to cellular proteins that actually act as motors, kind of like a car engine, and then pull hand over hand along these microtubules to deliver it down an axonal shaft. Here's an example of that red fluorescent virus doing this now live. Each one of those spots is an individual viral particle. It's one of those polyhedrons that I showed you at that super high resolution. Obviously, this is much lower resolution, but you get to see it happening now. And there's a couple things you can see right away, is that as these viral particles are marching in from the ends of the nerves to the neural cell bodies, they pretty much seem to know where they want to go. They're moving left to right, as they should. Once in a while, they'll pause, and sometimes you might have just saw that one. They'll actually reverse direction as if they got lost, but they quickly regain forward motion if that happens. That's what we call in the lab the marathon run, because it's not happening just over this little field of view, it's happening over a very long distance. And keep in mind that if it was you, it's entered here in your trigeminal ganglia, it's about that much distance, and to a virus, that's like running around the world in a straight line 3 times. It takes it about 8 hours to do that distance at the rates that it goes. If instead of imaging in the nerves, you image the neural tissue itself, we call this downtown in the laboratory. If you want to look at downtown, this is what it looks like. These are the neural cell bodies. There's those nuclei that we're talking about before. And this has actually been infected by these viruses, and you can tell because if you switch over to the fluorescent channel, you can see them. This is the finish line for the viruses. They make it to the nucleus, they dock on it, and they inject their DNA into the nucleus to take it over. And that's the process that's being hinted at here. This is 8 hours after, when you were 5, one of your grandparents kissed you, had a cold sore on their mouth. That was your ganglia, right there. Now, one other twist to this is that you can actually try to understand the composition of this particle as it's doing this marathon run by making it fluorescent different colors. And so in this particular recording, we have 2 different colors, green and red. We have the polyhedral capsid in red, and then we're trying to understand in these tegument proteins which ones transport with the capsid. And this particular protein that we've marked, you can see is associated with the red signal as it moves. Now, it kind of splits a little bit as it moves because in this old movie, we're taking one picture after the other, and by the time you take the second picture, the particles already moved a little. So you get this artifact that looks like they're separated, but it's really the same particle. And once we started understanding what proteins were on this large capsid, and we had some hints which ones might be the important ones for the infectious process. And this one that I'm showing you here was a big breakthrough for us. I don't have time to take you through all the details about it, but we discovered that this protein all by itself, when put into cells, would start marching around on these microtubule tracts hand over hand. And each one of these spots that you can see moving are moving on these ropes inside the cell, on these microtubules. This protein has the ability to do it all by itself. Nothing else from the virus is required. And once you have that information, you can start to tinker like crazy and make all sorts of changes into it to understand exactly how this process works. But what I want to finish with is I want to show you a much more recent discovery on a second protein. The names really aren't that important, but it's a protein that's not bound directly to the polyhedral capsid, but rather is bound indirectly through this other protein that I just showed you moving all by itself. And it turns out this protein, this accessory protein, is very important for the virus to infect the cell. And we've never been able to study it because if you delete it from the genetic code of the virus doesn't grow. And so there's nothing to study. But we did some structural analysis with a colleague in Boston named Katja Heldwein at Tufts, and she actually was able to determine one of these high-resolution structures of this specific protein. And when she did this, a picture's worth 1,000 words, and I know that picture probably doesn't look as cute as your grandkids, but to me, it's as beautiful as any. And you can see right here, that there are 3 regions on this protein surface, R1, 2, and 3. And these regions, because of her trained eye, she knew that these were conserved in all herpes viruses. She could predict that, and that those regions might be important, which means that we could now go in and target these regions and destroy them and see what ramification that had for a recombinant virus. And I'm going to tell you about one of them, region 2, which had no ramification. This is just looking at how much virus is being produced in a plastic dish with cultured cells in it over time. And you can see there's more and more virus being produced until it gets off and levels off in a plateau. And the important thing is that a healthy virus and our mutated virus behave basically the same. So it wasn't very interesting. That's that region here. But when you put this into neurons in culture, and in this case, we put them in a cage. We called it a chamber. And we put the neurons in such that the cell bodies were on one side, and the nerve terminals, the endings, the sensory endings, were on the far side of these channels. And we can now add the virus specifically to one side, and then image along these grooves, like between the prison bars. And here come the viruses doing their marathon run. They started off on the left, and they're heading to these neural cell bodies, and we're catching them in the act. And there's nothing moving here, despite that this virus was perfectly healthy in all other ways that we could see. The virus selectively seemed to be unable now to do the marathon run and get into the nervous system. That was the first time we've ever seen that process debilitated in such a way. We could also do this in a more crude, but sometimes more informative way. We could take them out of their cage, just have neurons out in the open, and just throw a bunch of virus all over them. And the thing that's interesting about that is it's a little messy. You see a lot of marathon runners here heading in, but there's a lot of stuff, these clumps of viral particles that never enter the cells. They're just sticking on the outside of them. But if you look at the ones that are moving, those are the ones that have gotten in and they're doing the marathon run. And the reason this is interesting to look at is that if you look at the mutant virus, you can now see what's going on. They get in and they start to move. They're on the microtubules and they're doing the hand-over-hand rope thing, but they're going both directions. They reverse constantly. And they never go anywhere. This is one way you can measure it. If you just start a movie and just take any one particle of your choice, say this is where it was when the movie started, here's where it is when it ended, that's how far it's moved, its displacement. And with a healthy virus, a wild-type healthy virus, you can see they're displacing, on average, within that time frame, 10 microns. So a short distance, it's a very short time frame. But that distance in the positives means they're heading in. When you look at our mutant virus, it's 0. We call this ping-ponging. They just go back and forth, and they never go anywhere. And the nice thing about that is that we now have something that we've wanted for a long time. In theory, a virus that was selectively unable to get into the nervous system, a lot of people predicted would be the ideal way to try to vaccinate against the neuroinvasive herpes viruses. And this was our first opportunity to find out if that was true. And so in this experiment, we gave, I should mention that herpes simplex virus, I mentioned if that gets to a rhesus macaque, it will cause encephalitis and kill the monkey. The same is true actually with a mouse as well. And I don't usually like to talk about animal experiments. I don't like to do them myself, but in some cases, you have to do it a little bit just to get to the point where you have an important product that you can use to save people. And this is one of those times, I believe. And so this is an experiment where we have not vaccinated the animals, we're just a sham vaccine. And what we're seeing is that the virus is getting into the peripheral nervous system and the central nervous system. So this is looking at infectious material that's made it to these tissues of a mouse. And when you give it first the mutated R2 vaccine, that ping-ponging virus, and then you challenge with a healthy virus, that healthy virus is not detected in the nervous system, neither the peripheral or the central. Your immune response to the vaccine, or the mouse's immune response, has made it completely resistant to the infection. And I just want to show you too, this looks bad, but it's really not as bad as it seems. This is kind of like the gingival stomatitis in the little kid. It looks bad. The mouse eye is getting swollen. In this case, the virus was inoculated on the eye itself, and it causes a swollen infection normally. But if you first give it the vaccine and then put the virus on the eye, you can see the eye looks perfectly healthy. And this is just a measure of that quantitatively. So the vaccine is preventing not only the ability of the virus to get into our nervous system, but it's also preventing the virus to cause any noticeable disease on the body surface. And this is my favorite experiment, because this has never been done before. People have been trying to make vaccines for a long time with mixed success. A lot of people will say, it's very easy to protect the mouse, Greg, but can you protect a human, and how do you know until you get there? But nobody's done this. This is a protection where you're looking at how well do mice survive a challenge of wild-type virus, and by 2 weeks, you can see they're fully surviving. Now, what we've done here that was different before is that we're not using herpes simplex virus. We're using a virus called pseudorabies. It's a very nasty neuroinvasive virus in the animal kingdom. Fortunately, it does not touch primates, including us. So it doesn't cause disease in humans, but this is one of the most nasty viruses known in this group. It goes straight to the brain almost every time. Think of it as like a monkey being a human going straight to the brain. And no one's ever been able to protect a rodent from an encephalitic infection. And we're getting 100% protection. And not only are we protecting them, but all you have to do is just put a little bit of that virus, that tame virus, on the body surface. No needle, no adjuvants, nothing aggressive, just a simple touch. And you only have to do it once. And within 2 weeks' time, the mouse is completely immune to this lethal infection. So in summary, what I'm showing you is that we can now take a system where the virus typically will replicate out in these peripheral tissues, gain access to nerves, and transport within the axons to the sensory neuron, where it establishes these lifelong latent infections. These infections can also cause complications, as we talked about before, by going into the central nervous system. And because the viruses are so good at doing that, they get there before your immune system can stop it, and they've already seeded our nerves before we know what is even happening. The challenge was to come up with a way to try to block that through a vaccination. And the breakthrough is that the invasive machinery that the virus is using is actually encoded in that tegument layer, those proteins that we have been talking about. And by altering those so that they lose the capacity to move the virus on those ropes over these long distances that are necessary for the marathon run, we now have a neutered virus That allows for good vaccination because within 2 weeks, all the virus is still out here, your immune system is fully kicked in, sterilizes the infection, and sets up protection for your nervous system for the future. And so, as mentioned in the introduction, we have started a startup to try to bring this technology to market. These are my colleagues, Gary Pickard and Katja Heldwein, did the three-dimensional modeling. Gary is a neuroscientist who helps look at the neuroinvasive process. And my last quote, I've always liked this quote, the implications are commensurate with the ambition. This is actually a quote that has nothing to do with what we're talking about here, but rather was a quote from Colonel Neil McCaslin, and this had to do with the Ronald Reagan era, and he was working on this program, the Star Wars program, to shoot down enemy missiles from space, and that's what he said about his program and why it should be funded, and that's not going to be our official tagline for us, but I kind of like to use it here as well. So I just want to mention the people who did this work. In particular, I want to mention a postdoctoral researcher in the lab who just recently moved to MIT. Her name's Alexia Richards, and she's right here sneaking her face into this group shot from the lab. And I've mentioned my colleagues. I also want to I mentioned Hangzhou one more time as well, who helped with that large structure of the polyhedron. And with that, I'd be happy to take any questions. Thank you. I think we have our first hand here. Yep.
Doctor, that's outstanding. I wanted to cheer back when you said that you killed that little devil. But for those of us kissed by our grandmothers when we were 5.
Could have been your father, but probably they're not as affectionate.
Let's keep it to your topic, Doctor.
Okay.
For those of us who have already inherited the herpes virus and it's alive and well and living inside,
yes.
What can you do for me?
So the best thing, of course, you probably know the answer, is acyclovir. It's an antiviral drug specific to herpes simplex virus and related viruses. And it's a wonderful treatment. It's particularly wonderful because there's an enzyme that the virus uses to replicate that's unique to this virus that this drug targets. And when it gets targeted by that enzyme by the virus, it turns it into an active form that kills the cell the virus is in and destroys it. And that means that it only will kill infected cells and leave all your healthy tissue alone without toxic side effects. And that's why doctors can give you acyclovir even at high dose and not have to worry about overdosing you and causing trouble. So that's a way to try to treat a recurrence once it started to occur. But as far as probably what you really want to hear, how can we go in with a missile strike and take it out of my nerves altogether? That's a tough one, because it is, by being in all those nerves, it's hard to imagine how you can get it out. I can tell you that some people had the recent idea, and it's showing mixed success, of trying to come in to your nerves with something that would cause it to turn on. So reactivate it, but reactivate it 100%. Every single nerve that's got it, turn it on. Start the virus is being made, and then come in with acyclovir at high dose, just at that moment, and kill every cell that it was infected. And in principle, that might work, although you could lose a lot of your peripheral neural perception as well. You might get numb, I would imagine, and there might be other complications. And it's also tricky to get all those things to reactivate so that they become targetable. When they're sleeping, they're very hard to see. Right here front.
That was a gorgeous presentation.
Thank you.
Are there any particular receptors on the nerve cells that are specific for the herpes virus? And if such, what's the normal function of them when you don't have the herpes? And the second part of the question is, why is the herpes so specific toward the middle part of the temporal lobe when we get the herpes encephalitis?
Okay, 2 very good questions. The first question was, what is the virus binding to in our cells that lets it to get in? And why is that protein there? It must be doing something else normally during our healthy states. That protein is, there's a couple, but the predominant one is called nectin, Nectin. And that's a protein on cells, not just nerve cells, but all cells of our body that are very important for cell-cell binding and communication between cells. The fact that that's what that protein is normally doing has nothing to do with the fact of why the virus has used it to get in. And this is true in all of virology. For every virus you can think of, there's something that the virus is targeted to bind to on the cell surface, and it usually at first glance, second glance, is very arbitrary. It just found something to use. It doesn't matter what it was doing normally. As long as it's in the right place for it to get in, it can use it. And this is what it happens to use in this case. The second part of your question was why, when those poor unfortunate individuals get an encephalitic infection, why is the encephalitic infection normally found in a part of your brain called the frontal temporal lobe? 90% of encephalitic infections with these viruses are just that. And in the frontal temporal lobe, 10% turn out to be in your brain stem. That's less known, but it's interesting because what the frontal temporal lobe region is receiving input from is your nose, olfaction, smell. You have the only place in your entire body where your nervous system is exposed to the outside world is right in your nose. Those neurons are binding to molecules that are odorants, and they're causing them to fire and gives you your sense of smell. And so the virus can touch those neurons directly without first replicating in any other tissues in your body. And so that's been a concern for a long time, that might be an easy entry portal. And in fact, with encephalitis that you're talking about, that's the best guess of what's happening, is the virus has somehow gotten into the olfactory nervous system and moved to the frontal temporal lobe. I should say that's speculation, though. We can't really prove that. It certainly does happen in mice, though. Brainstem is also very interesting because, and I'll just say it very quickly, if you get that rhesus macaque virus and you die of encephalitis, it's not frontal temporal lobe, it's your brainstem where it's going. And so there's something about us and our frontal temporal lobe and our olfaction that is heavily guarded. It's heavily guarded, so viruses won't usually get in. And we now know that there's a molecule called toll-like receptor 3. It's another aspect of research my lab's working on that's there specifically to work this détente with the herpes viruses, to keep it out of the central nervous system via that route, but to let it get into our peripheral nerves. That's the compromise that evolution has given us. The monkey virus has lost that ability, and it goes straight to the brainstem every time and kills you, and vice versa. So that's all part of what's going on in that homeostasis with this virus. Yes, right here.
Someone in my community in their 80s was very athletic. She exercised constantly, and she got viral encephalitis after playing 3 hours of tennis without hydrating herself, and went to the pool.
Yes.
And went swimming. So that's related to your comment about the olfactory going through the nose, and probably the water had the virus in it?
It's possible, although when you get to our age, chances are the person's already been infected long ago. And so it could be that in that particular case there was a recurrence, and for some reason the recurrence allowed it to get into the nervous system. I unfortunately just lost a colleague to this at Northwestern, a professor who does not study viruses at all, but had a sudden onset of herpes simplex encephalitis. And it's quite sporadic. You would never be able to predict it's about to happen. And it's unfortunately very hard to say what the cause and the effect are. Sometimes the observations that you just talked about could be causal, but they could also be just coincidental, and it's really hard to know. Sure.
Can you comment about Epstein-Barr relationship to herpes family and the cardiomyopathy is secondary?
Okay.
Also, there's a complication with acyclovir, and that's kidney stones.
I'm sorry, say that last part?
Acyclovir and kidney stones.
And kidney stones, sorry. Okay, so there's a couple things there. One, Epstein-Barr virus I did not mention. It's a herpes virus, but it's a herpes virus in a different subfamily. They're not neuroinvasive pathogen. It's not a neuroinvasive pathogen. Instead, it goes into the lymphatics. Of course, many of you know this is the virus in childhood that causes infectious mononucleosis, or mono, the kissing disease. It's also a cancerous virus, because one of the things it has to do when it gets into your white blood cells, or your B cells, is it has to be able to cause that cell to proliferate. And any time a foreign agent goes into one of your cells and says proliferate, that can later turn into cancer. And so Epstein-Barr virus is known for that complication. The other question was, is acyclovir, one complication you mentioned is that could cause kidney stones. Yeah, I was actually not aware of that, but thank you for mentioning that. But you suffer from that, huh?
We have time for one more question, and there's one right here.
Okay, and I should just say, if anyone wants to chat with me afterwards, I know we're running out of time, I'm happy to do that. Yeah.
That was a wonderful talk. Thank you. I have a million questions. I'll try and keep it down to maybe one or two. If I'm understanding the mechanism of your proposed vaccine, You've essentially disabled the R2 region, and it's actually gumming up the microtubules so that the new infecting viruses can't actually get to the nucleus anymore. Is that more
Pretty
or
close.
less?
I don't think that there is any kind of gumming up so much as that the virus no longer can do the hand-over-hand motion continuously over a long period of time. And so it just constantly lets go, and then sometimes it'll just grab the other direction, and it's doing both in perfect balance. It's random, so it can't go anywhere. But
So-
it's not blocking up, yeah.
When a new infective virus encounters somebody who's been vaccinated with your vaccine,
Yes.
what is the actual mechanism of blocking that virus from going upstream? And then if the virus comes out of latency, wouldn't that also, being treated by your vaccine, also prevent the virus from moving back down the axon out of latency?
Good question. So I'll try to make this quick. The vaccine, like any good vaccine, it should develop a type of immunity that's called adaptive. And so basically involves your white blood cells, your B cells, your T cells to come in. I kind of think of them as like the FBI that comes into the crime scene after it's happened. And so they're looking at all the death and destruction of all these cells that happened from this viral infection. And they're looking at all the pieces and gathering all the evidence. And they're putting together this portrait of who this invading person or virus is. And then they get a special crack team together and say, you hang out for the rest of this person's life for as long as you can, and you look out for this. And if you ever see this again, stop it before it even has a chance to get a foothold. So that's your adaptive immunity, and that's what you really want to have established. Infection in nature does that, right? But the problem is these viruses often, because they're so aggressive, will manage to do damage, or in this case, cause a lifelong established infection before that whole response can kick in. And the interesting thing for your final part of your question is, if you have this latent infection and it recurs, well, that native virus that infected you years ago actually caused that adaptive immune response in you. You have it. You have everything my vaccine could possibly do for you, the natural virus did. But unlike the vaccination process, the virus that did it to you got into your nerves. And when it's in your nerves, it has a number of resources at its disposal to prevent that adaptive immune response from killing it quickly. So basically, a battle ensues. It tries to stave off all of the defenses that your adaptive immune response is bringing in at it, just long enough to get on the surface of your lip so that you can kiss your friend. And it will succeed. But then eventually, that adaptive immune response will clear it again.
And let's thank our speaker.
Okay. Thank you.